This is our DNA model. We will be using it to explain the following concepts. So now let's start. The function of a DNA is to store an organism's genetic information and controls the production of proteins and is thus responsible for the biochemistry of an organism. It is often compared to a blueprint since it contains the instructions to construct other components of the cell, such as proteins and RNA molecules.
The sequence of amino acid residues in proteins is encoded in DNA using the genetic code. The actual genetic coding is due to the sequence of bases on the DNA strand. Every amino acid in every protein is coded by sequences of 3 bases.
The structure and function of the thousands of proteins in an organism is controlled by the order of the amino acids in the protein, and therefore is ultimately controlled by the sequence of bases on the DNA. Where is DNA found? In eukaryotes such as animals and plants, DNA is stored inside the cell nucleus, while in prokaryotes such as bacteria and archaea, the DNA is in the cell cytoplasm. These bases of a DNA do seem very important, don't they? But what exactly are they? To understand, let's first talk about what a DNA is made of.
DNA is made up of small building blocks called nucleotides, which in turn consist of an inorganic phosphate, a pentyl sugar, and a nitrogenous base. An inorganic phosphate and pentyl sugar will only give you a nucleotide. The phosphate group is derived from phosphoric acid and gives rise to the negative charges on the nucleotide. In our model, the orange fuzzy stick represents the inorganic phosphate group. In the pentyl sugar of the nucleotide, the carbon atoms are labeled 1' to 5'. The green fuzzy sticks in our model represent the pentyl sugar.
The nitrogenous base can be divided into purine, which consists of thymine and cytosine, as well as pyrimidine, which consists of adenine, guanine, and uracil, while uracil is only used in RNA. The nitrogenous bases are represented by the ice cream sticks. The red ice cream stick represents guanine, the yellow ice cream stick represents cytosine, the blue one represents thymine, and the purple one represents adenine.
Why is DNA a double helix? Now, the bases are hydrophobic, while the sugar and inorganic phosphates are hydrophilic, as DNA is located in a cell which is filled with water. So, how is DNA supposed to be in a cell when the bases are hydrophobic? Well, that is why DNA is a double helix. The double helix structure will mean that the sugar and phosphate group protect the bases from water, allowing DNA to exist in the cell. That is why our model is in the shape of a double helix. Notice anything strange about the way DNA is arranged? Yeah, the strands are not pointing in the same direction. They point in different directions. They are anti-parallel. One strand ends with 5' end, which is the phosphate group, and the other strand ends with a 3' end, which is the pentyl sugar, as represented in our model, where one strand ends with the green fuzzy stick and the other ends with the orange fuzzy stick. Also, the letters on the bases of one strand will be the right side up, while the letters on the other end will be upside down. So, how exactly does the phosphate, base, and sugar link up to form one nucleotide? Remember in biology last year, when we learned how when two glucose are joined together by something called condensation reaction, where one O, oxygen, and one OH, hydroxide, would be taken away from a glucose each, and water would be produced.
The nitrogenous base and the phosphate group each join to the pentyl sugar via condensation reaction. In the first condensation reaction, the nitrogenous base joins to the one carbon atom of the pentyl sugar with the elimination of water forming a nucleoside. The second condensation reaction, the phosphate group would join the 5 carbon atom of the pentyl sugar, resulting in a nucleotide. So these nucleotides would actually join up to form a DNA strand. The phosphate of one nucleotide is attached to the sugar of the next, resulting in a backbone of alternating phosphates and sugars. As can be seen from our model where the structure of the DNA alternates between the green and orange fuzzy stick. In addition, the bases would always be projected from the pentyl sugar. This is represented in our model when how the ice cream stick representing the nitrogenous base is always stuck to the green fuzzy stick, which represents the pentyl sugar. The polynucleotide strand has directionality from the 5N with the phosphate group to the 3N with the OH group. The nucleotides are joined together by strong covalent bonds called phosphodiester, which is represented by the black colored part between every 2 green and orange fuzzy stick. As you can see when the nucleotides join together, the nitrogenous bases pair up. These are the bases which we talked about earlier. Their sequence encodes information. The way the bases pair up is actually really specific. Purine only pairs with pyrimidines, so the total concentration of pyrimidines and purine are the same. This is shown in our model where the yellow ice cream stick only pairs with the red ice cream stick and the blue ice cream stick only pairs with the purple ice cream stick. Aniline only pairs with thymine through 2 H bonds as shown where there are 2 black lines connecting the purple ice cream stick to the blue ice cream stick. And guanine pairs only with cytosine through 3 hydrogen bonds as shown where there are 3 black lines connecting the red ice cream stick to the yellow ice cream stick. Hence the 2 chains are complementary.
But why are the 2 chains complementary? DNA constantly needs to replicate in order to pass on more information and create more proteins and cells. But of course, in the process of replication, surely you don't want any careless mistakes. DNA replicates semi-conservatively. Imagine the DNA strand as a zipper. When DNA is replicating, it unzips. New nuclear bases are attracted to the exposed end of the zipper, forming new DNA. However, because A only goes with P and C only goes with G, only a complementary base pair would be attracted. Hence, ensuring that the replicated DNA strand would be the same and thus ensuring the parent DNA strand passes on its biological information to the daughter strand.